So, hello guys and welcome back to Excel Share Academy. So, now we'll be discussing the last topic, that is the effect of pH, temperature, solvent on the growth of the bacteria. Okay, so solute and the water activity. So, water activity here it is represented by AW. Okay, now it is the amount of water which is available to organism. So guys, this also like how water activity is going to affect, right? So that is something related to your osmosis. So if you have got more water, the chances that the cell can become hypotonic, right? So the chances are that the bacterial cell might burst out, or if it is hypertonic also. In that case, the cell is going to shrink away. In both the cases, what happens is that the bacterial cell is gonna die. Okay. So this water activity completely depends on the osmotic effect. So it is the amount of water which is available to the organisms. Now, higher the solute, lower the water activity. Obviously, right? Solute molecules वहाँ पे आपके ज़्यादा हो जाएंगे. So water molecules जो रहेगा वो आपका कम हो जाएगा. So that's why we say lower water activity. Okay, now it is reduced by the adsorption to the surfaces. Water activity of a solution is 1/100th of the relative humidity of a solution. So what does it mean, this sentence? If you calculate the relative humidity of that particular condition of the environment, the water activity is going to be 1/100th of that particular relative humidity of the solution. Okay, now it is also equal to the ratio of the vapor pressure of the solution divided by the vapor pressure of the water. So that's how the water activity is given. This is just for the understanding, and the questions will never come from this particular topic. Don't worry about it. So solutes in the water activity. So already discussed like what is hypotonic and hypertonic. I'm not going it again. Now microbes adapt to changes in the osmotic concentration. So how they are going to adapt, that is what we are going to see right now. Able to follow, Krishita and Shruti?
Yes, ma'am.
Okay. So they reduce the osmotic concentration of the cytoplasm in a hypotonic solution. So that is because they have got the mechanosensitive channels in the plasma membrane. We have discussed about this, right? So whenever there is change, so this is like the mechanosensitive. So the if this is the bacterial cell, then these are present on the surface. Okay, so they understand that, okay, so the osmolarity over here is high and here it is low, because of which what will happen, if it is here high and it is here low, so the water will tend to come inside, right? But we don't want that. So in order to prevent that, then we have got these channels, mechanosensitive channels, so that it will not allow the excess water to go inside. Okay, or they can increase the internal solute concentration with compatible solutes to increase the internal osmotic concentration in case of the hypertonic solution. So is case mein kya hoga, when you have got the hypertonic condition, hyper, so in that case they try to take the solute from outside to inside. They try to maintain the balance of the outside solute concentration and the inside solute concentration, making sure that it is not going to result in the excess of water loss plus the precipitation of the proteins which is present in the cytoplasm. Okay. So they have got certain areas where they just keep it, so the small vacuole type organelles are there, so they'll keep the excess of salt over here, and it will be kept in one side of the cell, so that it just doesn't get shrink or dead because of the hypertonic conditions. Okay. Then we have got the extremely adapted microbes like halophiles, which can grow at high concentration of salt. Extreme halophiles, they can go till 6.2 molar ratio. So what exactly they do is that inside the cell, okay, inside the cell, they have certain, as a vacuole type organelles, in that they start accumulating the excess salt. Okay, and little salt will be taken so that the cytoplasm osmolarity and the outside osmolarity is little bit nearby, not exactly. See, ab kaisa hota hai that if you take show now. Okay, yes, I was explaining that. So inside a bacteria, you have got those vacuole kind of organelles which are present, and in that excess of salt they will be stored over here. So the storage can be in the form of crystals also. Okay, now extreme halogens, they can go again the higher concentration till 6.2 where it is able to survive. Okay, now do you think that when you say halophiles, it means if I try to grow in normal nutrient broth, or let's say LB broth, is it possible for these organisms to survive in normal broth, which has got a hardly 150 millimolar of salt concentration?
No.
Yes? No?
No. No, ma'am.
Correct. So they cannot survive in normal salt concentration, okay, because that has become their requirement now. So they require salt concentrations between 2 to 6.2 molar concentration. Okay? So those are about the halophiles. Now coming to the pH. So based on the pH, again, you can have acidophile and the alkalophile. So acidophile means where the pH is going to be very less, and alkalophile where the pH is going to be very high. Now, based on this pH, I have got a query for you guys. Can you tell me why pH ranges from 1 to 14? And why not from 0 to 100 or 20 or 50? Why it is 14?
That is based on hydrogen concentration, so which adds to the acidity of the solution. Laurie Bronsted.
Krishita, more like you have to brief your answer. You are on the right track though, but if you give it is because of the hydrogen concentration. It's like I say, hawa mein answer diya, right? So you have to be very specific.
So basically it's the hydrogen concentration, but then power is in raise to, no? So that is equivalent to like 100 folds.
So you are saying just because here it is 10 raised to minus 14, that's why we have got till 14. That's what you are saying?
I'm not able to explain it, but that's what we've studied in like 12th and all.
Yeah, so see, that's what the point is here. Though we have studied, still we don't know. Correct? It's not clear. We know that the pH ranges from 1 to 14. But why? So that's why ka answer, you have to make it clear. Okay, so this is your assignment. Tomorrow I'll ask. See, this can go 10 raised to minus 15 and 10 raised to minus 16. So it doesn't mean that ye aapka 15 ho jayega, ye aap pe 16 ho jayega. There is a reason. And yes, we all have studied. Now it's your job to get it clear. Because see, in future, whenever you're going to do, right? Interview to cross karna hi hai, even though you clear the exam. Clearing CSR is not a big deal right now. Okay. Everyone clears the exam. The only problem is with the interview clearing at a good institute. Abhi, with this knowledge, if you go to university, okay, easily you will get accepted. And I always tell my students, university ab last option rakho. Okay, always go for the institutes jahan pe sirf research chal raha hai. Let's say that somehow university you got, maybe good university. Let's say Pune University is good. You can do research over there. Delhi University is also good. So let's say that you have gone there. Okay, it's good. After Research Institute, if it is universities wise, then Pune and Delhi University is good. I don't know about the rest of the universities. Hyderabad is good. Hyderabad ko kaun sa hai wo ek central something is there. Central University, I think they say. This is good. Okay, now let's say that you have gone to the university. वहाँ पे ये extra additional काम रहता है that you have to go and teach your BSc and MSc students over there. Just imagine asking this question, right? So you should be able to answer that. So not a big thing. You can go through it and get the answer. Okay. So based on the pH, we have got the microbes which is coming under the acidophiles. Neutrophiles means जो आपके normal bacteria हो गए. Alkalophiles, which is like alkaline regions. So these bacteria, they maintain the internal pH near neutrality. Now acidic tolerance response, see again how they are able to do. So this also we have discussed. There is something called H plus ATPase pump. right? So this we have discussed earlier in cell membrane topic. So how this is going to do? So in order to maintain that the acidic condition, these ATPases, they use energy and they keep on bringing the H plus against the concentration. ठीक है? So ATPase यानी कि ATP यहाँ पे लग रहा है, आपका use हो रहा है. So they are the pumps actually which pumps the proton out of the cell. If see, this is the bacteria. यहाँ पे एसिडिक pH ज्यादा है, सॉरी ऐसे नहीं होना चाहिए, pH ज्यादा एसिडिक है, it means pH लेस है, right? Now this cell has to survive. कैसे करेगा? Either it will take the H plus or right, it will convert into something so that the surrounding pH is getting neutralized. ओके अगर उसके अंदर H plus ज्यादा है, that is pH is inside high and बाहर कम है, then it is going to pump out. Clear now? Now, otherwise the organisms is going to produce acidic or basic waste products. So जैसे carbonate हो गया है, the organic acids are there. So all these will be produced by the bacteria so that they can maintain their pH. If it is requirement of acidic pH, either they will take the H plus inside the cell. Okay, if it is basic, then they will try to pump the H plus out of the cell. Clear now? As such, not there anything in this, it's just the revision for you guys. Now temperature. So temperature sensitive bacteria are there. So thermosensitive, thermotolerant, right? So all these are there. Thermophile, okay? So they are the microbes which are able to survive at higher temperature. Now how it is possible? By synthesizing the enzymes which are like chaperones or heat shock proteins, which is ensuring that the proteins which is required for the survival of the cell is maintained, or it is prevented by getting misfolded or denatured at higher temperature. Clear? Now they exhibit cardinal growth temperature, minimum, optimum, maximum. So yes, we know at cold conditions the growth is getting delayed. Then we have got the optimum and then maximum, where the temperature is ideal for that particular organism to grow. So based on the temperature range, we have got psychrophiles, psychrotrophs, mesophiles, thermophiles, and hyperthermophiles. So this all I believe you guys know all this thing. One example is also given. That's okay. No need to remember or by heart because we have got so many organisms. ओके बट यस अगर मैं कल को ये पूछ रही हूँ कि ई कोलाई क्या है, साइक्रोफाइल है या थर्मोफाइल है या मीजोफाइल है, देन यू शुड नो द आंसर एटलीस्ट, राइट? सो दीज आर द मॉडल ऑर्गेनिज्म जो रहते हैं, बैसिलस हो गया, ई कोलाई हो गया. So this and all we should know. Now let's say that the DNA polymerase which we take for PCR, that is harvested from where? Which organism? It's a thermophile. What is the name of that organism? DNA polymerase used for PCR reaction?
Yeah. Thermus aquaticus.
Correct. Correct. It is your Thermus aquaticus. Good. Okay. So that's why it is also known as the Taq polymerase. I say Taq polymerase. Okay, good. Now, how adaptations of thermophiles? So there are like many ways by which the proteins can be more stabilized, by increasing the H bonds or having more prolines, or by synthesizing more chaperones. There is another advantage, like I have mentioned in one of the lectures also, like if the protein is having high number of disulfide bond, okay, then also the protein becomes very resistant to the temperature. Or you can have histone-like proteins which is stabilizing the DNA. Now, the membrane, how do you stabilize a membrane? So having more saturated. अगर आपका जो organism है, if it is in the cold condition, cold temperature, then we expect that the cell membrane is having more of your unsaturated fatty acids. Okay. Now, if it is at hot conditions or hot temperature is there, that time the cell membrane should have more of saturated phospholipids or the fatty acids. Now the reason is in order to maintain the fluidity. So cold conditions में क्या होगा? The more you have unsaturated, so you can make your cell membrane more fluid-like conditions. ठीक है? अब heat में क्या होगा? If you have more of unsaturated, there is an increase in the fluidity of the membrane. So this will result in loss of your cytoplasmic components. Okay, so that's why we don't have much of unsaturated when there is a higher temperature. So instead of that, we are going to have the saturated one. Saturated fatty acids. So that it can decrease the fluidity of the membrane, which got increased because of higher temperature. Clear, guys? Like when it is saturated and when it is unsaturated, why and
Yes,
all? Okay. Now, for example, archaeal membranes are more tolerant to the increase in temperature because they have got the ether linkages as compared to the ester linkages, what we have in the phospholipids. So we have ester linkages. Now, to make it more strong, the archaeal membranes, they have got the ether linkages. Coming to the oxygen concentration. So here, based on the oxygen, you have got the anaerobes, aerobes, then facultative, obligate. So all these kinds of category we have. So why oxygen is required? Because it is required in the metabolic process, and at the ATC, it is the final electron acceptor. So that's what we say terminal matlab final electron acceptor. Okay, which results in the formation of water then. So वहाँ पे आपको oxygen requirement है. Now, aerobe means the organism which is growing in the atmospheric oxygen, around 20% of oxygen is required. Obligate means it is definitely required. Okay. Anaerobe in the absence of the oxygen. Obligate means if these microbes are present in the presence of the oxygen, then they will die. So obligate anaerobe means definitely the absence of oxygen is required. Microaerophils, where the concentration of oxygen should be less. Facultative, they do not require oxygen but grow better in its presence. So it's not like hard and rule conditions for them, right? So it's okay even if it is there. Aerotolerant, they can grow with or without oxygen. So these are the categories you can see. And the how are we going to check the content, like whether this organism is requiring the oxygen or not. Okay, so we go for the enzyme assay, help of the SOD and the catalase. SOD is anyone, Shruti? SOD is which enzyme, Krishita?
Dismutase, it is a dismutase.
Superoxide dismutase, correct.
Dismutase. Yeah.
Okay. So now this is the oxygen, what happens, obligate aerobe. So whenever you have SOD and catalase is added, what happens, a bubble formation will come. Okay, so there you can see this oxygenic zone is coming and where the oxygen is not present. So it needs oxygen. इसका मतलब क्या, बैक्टीरिया आपके ना ऊपर आ जा रहे हैं. So obligate aerobe means definitely they need oxygen. So जो भी बैक्टीरिया रहेंगे वो आपके टॉप पर आ जाएंगे जहाँ पे आपका ऑक्सीजन ज्यादा प्रेजेंट रहेगा. Now facultative, they can, they can also survive with and without oxygen. Aerotolerant anaerobe, so they are ignoring the oxygen. So आपको टॉप पे ज्यादा नहीं मिलेगा. ठीक है. Now where the oxygen is toxic, it means the bacteria will not be at the surface. वो सारे के सारे आपके deep present रहेंगे. Microaerophils, where the less concentration can be used for the growth. Okay. Now, to understand all these things, you can have this enzyme studies, SOD and catalase. So, in the case of the presence of the oxygen, or prefer or need, you can see that both are going to give you the positive test. Okay. Now, ignoring the oxygen, catalase needs oxygen. So, you won't see any activity of the catalase. It is toxic, कुछ भी नहीं आएगा. थोड़ा सा oxygen है, so you will get certain activity of the enzymes. Clear? Is this experiment is clear? Yes, Krishita?
So catalase would require oxygen. Catalase would require oxygen. अच्छा peroxide वाला.
हाँ. Okay. हाँ. Clear? Okay.
Yes, ma'am.
This is the chamber, like where you can, actually see the basis of the different oxygen sensitivity. So oxygen is easily reduced to toxic products like superoxide, hydrogen peroxide, hydroxyl radical. तो इसके लिए हमारे पास एंजाइम्स क्या है? SOD and catalase. ठीक है? So superoxide dismutase and catalase is there. So this is your anaerobic chamber. As you can see, it's everywhere, it's covered. Okay. Now these are the bacteria. Plates, can you see? These are the bacteria, they are present. Now what they have done is, so they have added in the chamber, they have added the catalyst also, the palladium pellets are there, so that it gets activated, the release of the oxygen, and it is getting converted also. So in this gas generator, water is added to the chemicals in envelope to generate the H2, okay, and CO2. So carbon dioxide promotes more rapid growth of the microorganisms. H2 है और CO2 बाहर निकला है. अब जो भी यहाँ पे ऑक्सीजन रिलीज हो रहा है, ओके, सो दैट विल बी रिमूव्ड बाय द चैम्बर एक्टिविटी, बिकॉज H2 अपने गैस पैक से रिलीज कर दिया. That O2 will combine with the hydrogen and it will give you the water. ठीक है? This is the chemical reaction which is taking place with the help of your palladium, the catalyst. Okay, now the anaerobic indicator is there. So methylene blue becomes colorless in the absence of oxygen. So agar oxygen hai, then the color change will be there. So you will understand that this particular anaerobic chamber is not working for your anaerobic bacteria. Should I explain once again? Or is it clear?
Ma'am, explain please.
Okay, okay. See, this gas pack anaerobic chamber, it is to ensure that the anaerobic bacteria can grow. ठीक है? उनके growth के लिए है. Now, how do you ensure anaerobic system where you don't want oxygen? So there is a gas pack. It's a packet, okay? In this, the chemicals are there, which slowly is going to release hydrogen and carbon dioxide. This much is clear. Carbon dioxide is required for your organism to, grow. Now we have at this position palladium, which is acting as a chemical catalyst. Okay. Now the oxygen, which is released out by the metabolic activities of the organisms present in these agar plates, they are going to release the oxygen. But oxygen shouldn't be present for your anaerobic bacteria. Correct. So this has to be removed immediately. वो कैसे remove होगा? Because this oxygen is going to combine with the hydrogen released from the gas packet and become a water. Okay. This much is clear. And that's how they ensure that there is no oxygen present. Okay. Now there is a strip or indicator is present. So ये blue रहेगा जब तक oxygen नहीं है. It will change its color if there is an oxygen. And then you understand that, okay, your anaerobic system is not functioning. Now it is clear. If it is blue, it means your anaerobic system is functioning. Clear? So this is very difficult actually. I tried working on anaerobic bacteria and seriously, it is very difficult to maintain. Little leakage in the system. Okay, if suppose your lid is not proper, a clamp is not proper, it is not tightly packed, and all the oxygen is not being removed, it is really very difficult. Then the organism doesn't grow, and उनके साथ काम करना तो और difficult हो जाता है. So my as a personal advice, not to work on the anaerobic system. So this is another way where everything is kept inside, and यहाँ पे these are the two ways by which you put your hands inside. So यहाँ पे हमेशा आपका anaerobic condition maintain करके रखते हैं. ठीक है? This is hood only, gas pack anaerobic system. So यहाँ से आप दोनों हाथ डालोगे, it's like a gloves. So whatever you need, you can actually put it from this side. आप इसको यहाँ से add करोगे. Okay, see airlock for transferring materials into and out of the chamber. So it's not like everything पहले से ही रहेगा. You are going to work on the bacteria, right? So कैसे रहेगा? आप लोग plates लोगे, you need to transfer it to somewhere or something you have to do. तो आप यहाँ से अंदर डालोगे, ठीक है? Then you wear your gloves. ये इसके attach रहते हैं. आप हाथ डालो, यहाँ से उसको निकालो. Plates है यहाँ पे. कुछ काम करो. ठीक है? Now keep it in the incubator there itself. let them grow in the anaerobic condition. So when this one is happening, what happens, the chances are that the oxygen might also enter. Then you have got this, क्या कहते हैं इसको? I forgot. Something which will, you know, it's like a vacuum. I shouldn't say vacuum actually. It will just remove all the oxygen present inside this chamber, just because you need to transfer your materials, like जो भी pipettes रहेगा, media रहेगा, whatever it is. Okay. So when you transfer it from here and till here, sometimes oxygen might enter. So this is which will try to remove the oxygen. Okay. Same जैसे वो वहाँ पे gas pack system कर रहा था. Clear?
Yes, ma'am.
Okay, now pressure. I think this is the small topic only. So again, so pressure may jo kaam, which can survive, sorry, the organism which can survive at a higher pressure or low pressure, they comes under the barotolerant and the barophilic organisms. Okay, for example, the bacteria in the archaea which can live deep sea with very high hydrostatic pressures. So these barotolerant are the organisms which gets adversely affected by the increased pressure, but not as severely as non-tolerant organisms. And there are certain barophilic organisms which require or grow more rapidly in the presence of the increased pressure. Okay. See, because of this only, guys, you know, till now, even though we know that there are so many organisms reported, ठीक है, उसके बहुत सारे नाम and, you know, subspecies, species, genus, so many are there. Literally, like millions of bacteria are there, known. Okay, it is there in the databases. Still, we say that of those of the total, we know only 0.01% of the total bacteria, because ocean is still yet to be discovered. So many bacteria, so many organisms are there. It's just that we are not able to go into that deep level to get the sample and, you know, to bring it out. And kaisa rehta hai? Let's say that your ocean, nobody knows the bottom of the ocean, right? Nobody has gone till there. Jitna bhi hum log gaye hain, let's say that this is the surface of your ocean, and let's say that we have gone till here. Okay, so jo bhi yahan ka jo environment condition hoga, even if you take the suppose sample from here, you need to maintain this environment in the lab so that you can grow. यहाँ पे कितना प्रेशर है, यहाँ पे किस तरह के न्यूट्रिएंट्स मिल रहे हैं, and of course, see, if it is so deep, it means there is no sunlight. Okay, so these are the bacteria which does not need light, so they have to be grown in a dark place, having the same nutrition which is present in this particular region, plus that much temperature, plus that much pressure you have to maintain in the lab so that these bacteria can grow. Okay, so all because of these limitations, we haven't, you know, studied so many organisms, because it is difficult to grow them in the lab, and every bacteria cannot just grow in nutrient broth or agar media. Okay, so that's why we say that till now it's only 0.01% of known culture we have. Now coming to the radiation. So basically, if you see, like we use UV rays especially to kill the organisms, right? Believing that they are not resistant, they need to be then sensitive. Then only these ionizing radiations are able to kill them. For example, ionizing radiations like X-rays and gamma rays, they result in the mutations. Believing that they are sensitive, so the mutations will result in the death. Okay, so because these mutations will result in the DNA damage, if it is not repaired by the repair mechanism, then the cell is undergoing the apoptosis or the cell death. For example, Deinococcus radiodurans, this is extremely resistant to the DNA damage. It means that it can just tolerate any, we say gray unit. In case of radiation, we say gray, right? Gray. Like 1 gray, 2 gray, 10 gray. This is a unit. So that much high level of radiation this organism can tolerate without getting killed. Now coming to the UV, so we know that DNA is absorbing UV radiation at 260 nanometer. Okay. Now, if it is continuous and if it is like very intense, then it can result in the mutations which will cause to the death. For example, causing the thymine dimer in the DNA. And if it is not repaired, then of course the cell is gonna not survive. Then visible light. If you put a very strong, intense monochromatic light, they will result in the formation of your singlet oxygen, which is a very powerful oxidizing agent. So when they combine with the proteins which are present in the cytoplasm, the organism loses its activity. Okay? For example, carotenoid pigments are there to protect the organism. तो ये जो सिंगलेट ऑक्सीजन जैसी चीजें प्रोड्यूस होती हैं वो आपके कैरेक्टोनॉइड्स होते हैं, xanthophylls होते हैं. They're like, you know, oxidating, anti-oxidating agents. So when they will absorb all these things so that your normal proteins, normal DNA is not getting affected. Clear? Even we say that the wrinkles, like in case of humans, wrinkles starts, right? Because after a certain age, you will see that our mother, father, or let's say nana, nani, dada, dadi, they all are starting getting wrinkles. Why? Because see, every time we get exposed to these radiations, light, so all this generation of the singlet oxygen and mutations that are happening to the skin cells whenever this radiation the person is getting exposed. The only thing was like earlier at the younger age, your cells were able to produce the enzymes, or you know, they had these antioxidizing agents were present so that you can repair your cell and the wrinkles were not seen. But later on, what happens? Your cell is losing its integrity. Cell is also tired of dividing. As I said, each cell will divide only max to max 40 to 50 times. After that, it will stop. So that stoppage has resulted in the reduced activity of the enzymes, which helps in removing these oxidizing agents. Okay. So because of this, one of the outcome is getting the wrinkles. So wrinkles kya hai actually, if you see, if there is a face and we say the wrinkles are coming like this, there it is the loss of the cells, and that's why those lines are seen. Those lines are nothing but the loss of the cells in that particular regions. Okay. Yeah. Now coming to the biofilms. So biofilm related, I have not seen any questions, but it will definitely cover the part of your microbial physiology. I hope everyone knows what is biofilm, right? So especially in the mouth, in the teeth plaque and all, these biofilms are formed, or in the pipelines where continuous water or the drainage system is there. So there we see the biofilm formation. So I've worked on biofilm formation and really it's very risky. First of all, you're working on pathogenic bacteria. Okay. Any mistake, you might get the infection. Then making biofilm, just to see that my drug is able to disrupt the biofilm or not. So even I have worked on this anyways. So the most bacteria, they grow and attach to the surfaces rather than, you know, free floating. So these attached microbes are the members of the complex slime enclosed communities called the biofilm. They're ubiquitous in nature and it can be formed on any conditioned surface. It's not like ab jitna zyada chikna ya plain surface rakhoge, the biofilm is not going to form. It will definitely form. Okay. Any idea ये क्या चीज दिखा रहे हैं यहाँ पे? Shruti, can you make a guess what is it? Krishita?
Actually, I was disconnected, ma'am. So,
probably bacteria encased in biofilm.
Yeah, but ये क्या चीज है? कहाँ पे है?
Oh.
No? This is heart. Heart.
Oh.
Okay. They have made a cut over here. This much deep they have gone, and they have, you know, what to say, the pacemaker is there. On the top of it, in the heart, this biofilm is formed.
This actually looked like a muscle. So how would you differentiate between a muscle and a heart if I had to?
No, then you can't. With this image, you can't. I know, I have taken the image, that's why I know it is heart.
Okay.
Otherwise, you can't. Otherwise, you can't. Okay, now how does this biofilm is formed? Okay, so now how the biofilm is formed? So first thing, first the bacteria, okay, it will see whether, all this thing is available or not, nutrition is available or not, whether it is able to secrete out the polysaccharides, proteins, okay, like the extracellular polymeric substance. This is something which is very difficult by many of the drugs to cleave actually, so that it can go inside. So biofilm mein kya rahega? You have got the surface. This EPS is actually covering the biofilm. It means the bacteria are inside. This is extracellular polymeric substance. And this is something which is protecting the bacteria. So we always needed a anti-biofilm agent which can actually disrupt this EPS. And this is like very sticky, very, what to say, I don't know, like it's very hard that you are not able to remove. And जब तक इसमें आप पोर बनाओगे नहीं, इसको डैमेज नहीं करोगे, inside the bacteria is there. You cannot kill those bacteria then. Okay. Now, suppose let's say the bacteria is there, substratum preconditioned by the ambient molecule. So you have got a very good amount of the food and the nourishment got by the bacteria. So then the cell will detect that and they will try to get settle over there. Now, as we have discussed in the case of the animal cells, those extracellular proteins are there and they make interactions with the extracellular matrix, right? In the same way, on the surface of these biofilm forming bacteria, let's say Pseudomonas aeruginosa, it's one of the example, they have got these surface based protein molecules. So they get attached on the substratum. Okay, so there the cells will try to, get adsorbed over here. Now, what is happening? It's not that every time the same bacteria will get attached and, they start forming. कैसा होता है? Community form होती है धीरे धीरे. So as you can see it over here also, 2 types of bacteria they have shown. So not necessary. So these bacteria need to communicate with each other. Okay. They need to communicate. If they are from the different species, they need to communicate with each other. And for the communication, they have to release certain molecules. Okay. So we'll discuss this in quorum sensing later. So what is happening in the biofilm formation is a quorum sensing. So they are communicating with each other within the same species and with also the different species. So they are communicating. So as you can see, ये यहाँ इसी भी discuss कर रहा है, ये इसी भी discuss कर रहा है. तो cell to cell signal हो रहा है, and eventually they are resulting in the release of the EPS. Clear? Now what is happening? They start growing it inside of this EPS. They replicate, they grow, and they keep on secreting out the matrix so that a protective layer is made. तो पहला ये आपका thin रहता है. As the bacteria starts growing, they all keep on secreting and it is attached, deposited over here so that it becomes very thick and very strong. So what happens is that even though, if suppose this is a pipeline, even though if water is coming with so much of pressure, it can just pass through. This will be still stuck to the pipeline. Okay, ये आपका कहीं नहीं जा रहा है. They are that strong. Unless until you come with a knife or something, to just say scratch it out. Otherwise, it is really very difficult to remove with just putting the pressure of water or some chemicals are added. This layer is so strong, it's so thick that none of the chemicals are able to penetrate and kill the bacteria. Okay, so it's actually a biggest issue in the medical field. Just say, for example, in the case of the heart, the pacemaker is there. If it is not sterilized properly, then let's say even a one bacteria is there, or maybe that person got infection. So they go and those area, those stent where you put in the case of the cholesterol people, like the cholesterol blockage is there, bypass surgery is there. So the doctor puts stent. All these are not going to react with the blood components. But if you have a bacteria, then they get settled over there. Or blood is what? It is like, unlimited resources, unlimited supply of the nutrition. जहाँ पे इतना glucose आ रहा है and all the other proteins are there, right? So what happens? Then they start growing over there on the medical implants. And that is the biggest issue. तो इसलिए कैसे रहता है, initially they have to keep on taking the antibiotics. In case they form a biofilm, then definitely that particular medical implant is not going to function properly, and plus the entire body will get infected. Okay, so it's a major issue in the medical field. Now, the EPS, the extracellular polymeric substance, and the change in the attachment organism, it protects the microbes from the harmful agents, let it be UV light, antibiotics, or the microbials. So when they are formed on the medical devices such as implants, often lead to illness. Now, sloughing of organisms can result in contamination of water phase above the bounds, such as drinking water system. So as this also we know, you can see that sometimes these starts getting collected. They start forming a deposits on the pipelines where the water is there. So this can be your drinking water, and that's why we say once in a month, whatever your aquaguard is there, even that needs to be cleaned. Otherwise, they start getting deposited. Okay. Now, the heterogeneity. Now, why heterogeneity? It is because it's not only a single organism. We have got different types. Okay. So a mature biofilm is a complex dynamic community of microorganisms. It is because the difference in the metabolic activity and the locations of the microbes. So har ek ka apna ek, it's like, what to say? It's like Mumbai. Like people are coming from around the world. Okay, not only from the India, but bahar se bhi hai. And they come and they get settled also sometimes. So the exchange takes place metabolically, DNA uptake and communication. So see, this is something like the exchange of DNA is also very easy. If suppose one bacteria is there which is resistant to, let's say, the antibiotics, right? Now it is a part of biofilm. So kya hoga? It can release those particular plasmids, right? Or those signals, and the rest of the bacteria can take those gene, okay, DNA uptake and getting transformed. So within a biofilm, it means that all the bacteria are going to be resistant for that particular antibiotic. Yes, did you get it? Is this point was clear, like how one bacteria can make the entire biofilm colony resistant towards the bacteria? Resistant towards the antibiotics? Yeah, okay. Now look it over here. So there are certain slow growers and the fast growers are also there. So kaisa hoga inke wajah se? Slow growers means they will take less of the nutrition. But if I have fast grower, they will take maximum of the nutrition, and it will be dangerous for the community, right? But they will also release certain signals in such a way that they recruit, or the more bacteria towards the biofilm, bringing the resources. Okay, so there is always change in the physiology. There is always change in the number of the organisms. So one is helping the other, giving the signal to come. Okay, now since it is EPS, extra polymeric substance, it is negatively charged. Okay, so based on this knowledge that your EPS is negatively charged, scientists have made antimicrobial, which is going to be positively charged. So that the interaction can be there. But unfortunately, like as far as I know, there are many substances which is not able to disrupt the biofilms. Okay, so killing the biofilm forming bacteria is actually difficult. Okay. So guys, that's all for microbial physiology. So here I say that the microbial physiology is completed.